Hello everyone. I'm honored to present our work on rapid reconstruction of multi-drug-resistant bacterial genomes using CycloneSEQ nanopore sequencing. Multi-drug-resistant bacteria typically possess complex genome architectures that harbor mobile genetic elements such as plasmids, integrative and conjugative elements, integrons, and transposons, as well as abundant repetitive sequences including insertion sequences. As a result, conventional short-read sequencing cannot resolve the fine-scale genomic organization (MDR) bacteria, hindering genome-level, high-resolution analyses of the horizontal transfer dynamics and dissemination risk of resistance genes. Recently, Chinese MGI launched CycloneSEQ, a domestically developed nanopore sequencing platform capable of producing single reads longer than 100 kilobytes. These long reads can span hundreds of sequences, enabling complete resolution of complex genetic structures common in MDR bacteria, such as plasmids, transposons, and tandemly repeated resistance regions, and thereby greatly improving the efficiency of MDR genome reconstruction. In this study, we resequenced 10 multi-drug-resistant strains spanning diverse species and sources using the CycloneSEQ to evaluate its capacity to reconstruct their genomes. We found that most CycloneSEQ reads were 10 to 20 kilobyte in length, with a subset of ultra-long reads more than 100 kilobyte. Analysis of per-sample read length N0 showed that most samples clustered around 8 kilobyte, ZN2 had the highest, N50, whereas IC72 had the lowest. Because nanopore library preparation does not require PCR amplification, we observed no obvious sequencing bias across the test samples, and each bacterial genome exhibited uniform coverage. However, when we assessed assembled genomes generated under different assembly and polishing strategies, we found that long-read-only assemblies produced numerous gene prediction errors. Consequently, obtaining more accurate MDR bacterial genomes requires short-read-based polishing or a hybrid assembly approach. Sequencing depth profoundly influences the completeness and accuracy of bacterial genome assembly. To evaluate the impact of CycloneSEQ sequencing depth on assembled genomes, we selected the strain with the highest coverage, randomly subsampled its reads to a range of target depths, and assembled each dataset independently. Using long alone, a complete bacterial genome was recovered at just 15-fold depth, with an accuracy of 98.88%. Assembly accuracy improved with increasing depth but showed no further gains beyond 50-fold. Under a hybrid assembly strategy, a complete genome was obtained with as little as 5-fold long-read coverage, indicating that incorporating CycloneSEQ nanopore data substantially enhances bacterial genome assembly quality. Multi-drug-resistant bacterial genomes often contain complex structures that conventional sequencing technologies cannot resolve. In particular, heterogeneous tandem repeats hinder accurate genome assembly and are closely linked to resistance phenotypes, because copy number variation within tandem repeat regions can alter the expression of antibiotic resistance genes and the stability of mobile genetic elements, thereby affecting bacterial resistance and the dissemination of resistance genes. In this study, CycloneSEQ was shown to effectively resolve tandem repeat heterogeneity of resistance genes in resistant bacteria. Recently, CycloneSEQ has upgraded its sequencing chemistry and flow cells, markedly improving accuracy. The latest CycloneSEQ platform achieves an average per-read accuracy of 96.16%. We found that genomes assembled using CycloneSEQ data alone showed no misassemblies and only a small number of base mismatches, yielding an overall accuracy of 99.98%. These results indicate that using CycloneSEQ data alone can potentially produce complete, highly accurate bacterial genomes without short-read-based polishing. Looking ahead, further improvements in library preparation chemistry, base calling algorithms, and cost reduction strategies could enhance the platform's practicality and expand its applications in microbial genomics and clinical microbiology. In summary, our study provides a comprehensive evaluation of the CycloneSEQ nanopore sequencing platform for MDR bacterial genome assembly. We demonstrated that CycloneSEQ long-read data enable the reconstruction of near-complete bacterial genomes, resolve complex genetic structures, and improve the quality of hybrid assembly. Using the CycloneSEQ sequencing platform, complete and highly accurate genomes of multi-drug-resistant bacteria can be assembled without the need for short-read-based polishing.